acuerdo.
Venga. Good afternoon. First of all, thank you to all of you for attending this webinar. Today we have the webinar from the group of mitochondria, cell communication and oxidative stress. This group is led by Susana Cadenas. Thank you, Susana, for organizing this webinar. And please, if you want, you can introduce our speaker today.
Yes, thank you very much, Mar. Good afternoon, everybody. It is my pleasure to introduce our speaker, Eduardo Balsa. Eduardo did his PhD at Hospital Universitario de la Princesa, where we first met and collaborated. He was working on metabolic adaptations to low oxygen tension, and he obtained very interesting papers in top journals. He also performed a stage in the laboratory of José Antonio Enriquez at CIPF, and then he moved for a postdoc at the Dana-Farber Cancer Institute and Harvard University in Boston, to the lab of Pere Puigcerver, where he did a brilliant postdoc. In 2020, he joined the Centro de Biología Molecular Severo Ochoa with a Ramón y Cajal contract, where he leads the group mitochondrial dysfunction in metabolic diseases. His talk today is about mitochondrial function as a gatekeeper for redox homeostasis. So, Eduardo, thank you very much for accepting our invitation, and the floor is yours.
Thank you so much. I'm gonna share the presentation. Again, thanks Susana, thank Maria for this kind of invitation. It's a real pleasure to be here. Today I'm gonna share you some of the data we've been working over the last years, which basically we try to uncover new mitochondrial functions. We try to debunk this whole idea of mitochondria and its link to bioenergetics. Mitochondria are not just the powerhouses of the cells; they do many more things. And we're investigating, these kind of things and how they link to mitochondrial disorders and disease. So you probably are very familiar with mitochondria, these small organelles within the cell. You can really hear in this electron microscopy picture. You can appreciate this tree state where this molecular machinery called the oxidative phosphorylation system is located. Which is this molecular machinery inside our cells, inside the mitochondria, that transform nutrients into energy. But as I mentioned, mitochondria are multifaceted, and they are involved in many biological processes, from producing energy from ATP, but also they're involved in many anabolic pathways, pathways that transform and produces different macromolecules. Also, there are important organelles in cell signaling that are mediated through reactive oxidation species or calcium homeostasis. They also involve in cell death and apoptosis and many other things. So they're important organelles. Of course, when there are mutations, both in nuclear or mitochondrial encoded genes that negatively impact the electron transport chain, that are the roots of a great group or heterogeneous group of metabolic disorders that we commonly refer to them as mitochondrial diseases, right? Those diseases can impact one or many organs, and their onset is quite variable. It can affect children, but also adults. Sometimes the affectations are mild, involving just weakness or intolerance to exercise. But there are more severe forms of mitochondrial diseases that are incompatible with life. So there are many efforts by our group and many others to try to uncover the molecular mechanism and try to develop novel therapies. So in terms of as I mentioned, they are quite heterogeneous. Also, that's what makes them really difficult to diagnose. As you can clearly see in this picture, the symptoms are quite high. But there are one common characteristics that occurs in many of these patients that are increased athletic lactate. At the molecular level, why it is happening? Well, when you have like a cell, a muscle cell, or a brain cell, or a heart cell with intact mitochondria, normally glucose is uptaken and is fully oxidized in the tissue cycle, right? And this is driving ATP generation. But what happens when mitochondria is not working? The consensus is that glucose is redirected towards lactic production because you want to increase glycolysis, and as a mechanism, as a compensatory mechanism, to keep producing ATP, because of course the electron transport chain in the mitochondria is not working, it's malfunctioning. Actually, like lactate accumulation in blood of this patient is a really good biomarker for the disease and disease progression. Something that drew our attention when we wanted to recapitulate that this phenotype occurs in vivo, and you can clearly see like two widely common use cell lines, killer cells and U2S. Those cells, they grow mostly fine, even in the absence of glucose. But when you target the electron transport chain, we did it with this specific drug that targets the complex one, you see that as glucose is being restricted, those cells start dying, right? How can we interpret this data? Well, according to the current dogma, it's because cells whose mitochondria is not working, they uptake glucose, they increase this glucose uptake, and they run it through glycolysis to produce ATP, right? But this leads to a question, which is like, okay, glucose is highly used, you need like glucose for these cells with malfunctioning mitochondria, but is there any other glucose-dependent pathway that is also essential for these cells when mitochondrial dysfunction? And this is the question, the fundamental question that was driving this project. So what we did is that we generate different cell lines with specific knockouts for metabolic pathways that we know that glucose is important. For instance, the pentose phosphate pathway, apart from glycolysis, of course, the pentose phosphate pathway, isosamine pathway, and the serine biosynthesis pathway, and also like the import of mitochondria into the pyruvate, sorry, import of pyruvate into the mitochondria through the mitochondrial pyruvate carrier. And then once we generate knockout cells for these specific metabolic pathways, then we start targeting the electron transport chain with different drugs, targeting complex 1 or complex 3, as you clearly see here. And then surprisingly, cells which do not have active pentose phosphate pathway, they were synthetic lethal with mitochondrial dysfunction, right? And this was quite novel. Actually, what we found is that when you inhibit mitochondrial function, it's not just like lactate is being released, or is increased, and this is a proxy of glycolysis, but also the pentose phosphate pathway activity increases as well. And this was demonstrated in primary neurons. And of course, normally you culture cells in 25 millimolar glucose, so they have like plenty of glucose. Those two pathways, the pentose phosphate pathway and glycolysis, they both depend on glucose, so they should compete for glucose, but glucose is in abundance, right? So what would happen if you restrict glucose? You go through a more physiological glucose levels, which is like 5 or 2.5 millimolar glucose. What you can see here is the opposite. It's quite dramatic. So at 5 millimolar glucose, there is a trend towards decreasing pentose phosphate pathway at the expense of maintaining lactate release, which is a proxy of glycolysis. And if you even lower down a little bit the glucose, you clearly see that the pentose phosphate pathway activity crashes. And again, it's at the expense of maintaining lactate release. So what we think is that when cells have to decide whether they want to run glycolysis or CPP, they favor the glycolysis, right, in the context of mitochondrial dysfunction. And you can clearly see later they'll have like huge implications. So what does the pentose phosphate pathway do? So the oxidative branch of the PPP, for short, is involved in generating NADPH, and this reduced molecule is important for many biological processes. It's important for reductive biosynthesis, of course, oxidative defenses, also detoxification. So then we perform a kinetics of these cells at different points, inhibiting just mitochondria, just PPP, and then both. And you can clearly see that beyond 48 hours, cells, where you inhibit these two pathways, you start clearly see this synthetic lethality, this cell starts crashing. And at those time points correlate with decreased intracellular levels of NADPH. Of course, because NADPH is important to detoxify reactive oxygen species, we start seeing increase in ROS levels. This cell death could be ameliorated by supplementing cells with glutathione. So we think that what is occurring is that mitochondria is important to generate NADPH that combat reactive oxygen species. So when the mitochondria is malfunctioning and you cannot compensate through the PPP, cells start crashing because of this phenotype. So what are like the pillars of like the main sources that produces NADPH within the cells? So mainly there are like three of them. The first one is these two enzymes, G6PD and PDGD. Those are involved in the oxidative branch of the pentose phosphate pathway, but also malic enzyme and the isoteric dehydrogenase. They're also important sources of NADPH in the cytosol. So what we did is, okay, can we express one of these two enzymes, and this, in theory, should counteract these deleterious effects. Surprisingly, when we express malic enzyme 1, this could counteract like this dual inhibition of mitochondrial dysfunction and PPP. IDH did it not at the same level, and the explanation for that is that when you inhibit the mitochondria, there are like certain metabolites within the cell that alter. Some of them accumulate, some of them decreases, because as I mentioned, mitochondria is also involved in anabolic reactions. So what we found is that when you inhibit complex 1, for instance, isocitrate, which is the substrate that the IDH enzyme uses to produce NADPH, is hugely reduced. So it's almost depleted within the cell. Whereas malate, the substrate malic enzyme 1, increases. So what we think is that somehow cells are able to adapt to mitochondrial dysfunction and keep producing malate through alternative pathways, and this malate is being used by the malic enzyme to maintain NADPH levels at check and also reactive oxygen species. So obviously the next question was, okay, like how do cells manage to produce malate even in conditions where mitochondria is not functioning? So normally malate can be produced from TCA cycle intermediates under normal conditions where like mitochondria function is intact. Glucose is transferred to pyruvate that enter the mitochondria and fuels the TCA cycle, right? And then, for instance, oxalacetate can give rise to malate in the cytosol to be used for the malic enzyme 1 and produce NADPH. But when mitochondria is broken, let's say, we speculate that could be alternative pathways, like adaptive pathways that are being upregulated and could maintain malate levels. One of them, derived from glucose, is through the action of pyruvate carboxylase. So pyruvate could still enter the mitochondria, be carboxylated from oxalacetate and then malate. And the other one comes from glutamine. Glutamine can follow reductive carboxylation within the mitochondria, give rise to citrate, and the cytosol through the ACLY enzyme be split into oxalacetate and acetyl-CoA, also giving rise to malate. So how can we test whether these pathways are more active or not in the context of mitochondrial dysfunction? So to do that, we took advantage of isotope tracing, stable isotopes, for instance, glucose. So this is the case I'm going to show you. So we generated knockout cells for PC, and what we did is incubate these wild-type cells or PC knockout cells in the presence of labeled glucose. This is fully labeled glucose. And we follow the trace of the glucose-derived metabolites within the cell. You can clearly see that if pyruvate dehydrogenase pathway is being followed, you end up generating malate, like molecules of malate that has these labels or these carbons label, right? We call it malate N+2. Whereas conversely, if pyruvate carboxylate was the fate under these conditions, then malate N+3 would be generated. So what we found is that when you inhibit mitochondria with pyridine, this is the blue bars, of course, the oxidative pathway, like the N+2 isoforms, are being depleted, right? You see like this huge decrease. Whereas this N+3 malate and all other N+3 metabolites have been increased, meaning that when you inhibit mitochondria, the fate of pyruvate is not going through the pyruvate dehydrogenase, but is being carboxylated by PC and then give rise to malate. And probably this is why these cells keep maintaining the intercellular malate levels even when the TC cycle is being affected. But surprisingly, those cells, like PC knockout cells, they proliferate fine when we target mitochondria. So that wasn't something that we expected. Alternatively, we tested another hypothesis. Okay, maybe it's the glutamine pathway. And what happens under normal conditions is glutamine is being oxidized in the TC cycle and create this malate N+4 metabolites. And again, we have like ugly knockout cells. And then when we inhibit mitochondria, the glutamine also follows reductive carboxylation and end up rising to malate N+3 isoforms. And then we can follow that. Again, we see that when we target complex 1, the N+4 isoform goes down. And again, this N+3, this alternative route is being upregulated and it's ugly dependent. But again, if you have these cells and you target mitochondria, the ugly knockout cells that, these cells shouldn't have like this pathway active, again, they can still survive. So what we thought is maybe like both mechanisms work together. And then when we target one of these mechanisms, the other is further upregulated. So how can we test that? So again, when we did these tracing experiments using glutamine in PC knockout cells, and you can clearly see that in those cells, when you inhibit mitochondria, the N+3 forms are upregulated. But if you further knock out the alternative pathway, the PC pathway that comes from glutamine, this is even more upregulated. So we think these cells can adapt. And actually, when you create these double knockouts, ugly and PC knockouts, it was you start seeing the defect. Those cells can handle transport chain disruption. So in summary, what happened is that both glucose and glutamine fits into this pathway, and you have like these two enzymes that transform these metabolites into malate. If you delete one of them, the other is further upregulated, the other way around. So when you target both is where you start seeing this huge phenotype. So of course, we have done these experiments in a couple of different cell lines. And the big question is that, okay, is this a general effect? It's something that, like a fundamental process in biology. So to test that, we analyze hundreds of cell lines. And we stratify or categorize these cell lines by the malic enzyme expression, which now is the key protein for cells to be adapted to these conditions. And then we obtain a couple of cell lines that were like malic enzyme high, and the others malic enzyme low. And then we tested our hypothesis. Well, the hypothesis was, okay, like malic enzyme high cells should be very insensitive to drugs that both target PPP and mitochondria, right? And the ones that are low in terms of levels of malic enzyme should be very sensitive. Actually, that's what happened. You can clearly see, and this is like viability test, like counting cells. Cells with high levels of malic enzyme, they don't care. You can target pyridine and pentose phosphate pathway at the same time. You culture them, they proliferate fine. Whereas the malic enzyme low cells, they crash rapidly. And that correlates with the levels of NADPH. Those cells can still maintain NADPH levels intracellularly, but not the malic enzyme low. Because those cells are quite heterogeneous in terms of the genetic background, we confirm that this phenotype is due to malic enzyme 1. We knock out malic enzyme in the malic enzyme high expressing cells, and then we see like the phenotype again, and then we overexpress it in the malic enzyme 1 low cells, and we still like we saw nice rescue. So the phenotype was attributed to the levels of malic enzyme 1. So we did another prediction again. Okay, like we can stratify cells based on the levels of malic enzyme 1, or also like glucose-6-phosphate dehydrogenase, like enzymes that produces NADPH within the cell. And we think we can predict whether those cells are going to be sensitive or not to drugs that disturb the electrotransport chain. So we obtained this cell line that was very low on the PPP pathway and also on malic enzyme pathway. And what we did is xenograph experiments. So we treated mice with a drug that also targets complex 1 and can be used in vivo. And you can clearly see phenocopying what we observe in cells, that mice that were given this drug, cells like the tumors that these cells form were really small. And then if we express again malic enzyme 1 in these cells, this phenotype completely disappears. So this we confirm in vivo what we also saw in these multiple cell lines. Of course, we were interested not in curing cancer, but curing mitochondrial diseases. And normally what happens is that there are like certain organs, mostly oxidative organs, such as the heart, the skeletal muscle, and mostly the brain that are affected, right? Curiously, the brain is quite heterogeneous tissue. You know, it's made up by many different cell types. So what we thought is, okay, maybe not all these brain cell types are equally sensitive to disruption in the electrotransport chain. Based on protein expression analysis, you see here like the expression levels of malic enzyme 1 versus the pyruvate carboxylase, and then you can clearly see that astrocytes have like high levels of both enzymes, and for instance, neurons have very low levels of these two enzymes. So based on that, we would predict that in astrocytes, during inhibit mitochondrial function, they will be mostly fine, but neurons will be very sensitive. And actually, that's what we saw. You treat neurons with pyridine, you can clearly see that they start dying, and we see like massive cell death after 24-48 hours. But you treat astrocytes with different poisons that target mitochondria, and they're mostly fine. They don't die, they proliferate, they're okay. Again, we validate that astrocytes do have like really high levels of these metabolic enzymes. They are active. We measure PC activity and malic enzyme activity by mass spectrometry, and it's almost triple like the activity that the astrocytes have compared to the neurons. And something that we found like really interesting, you target mitochondria in astrocytes, there is no drop in ATP levels or NADPH levels. Whereas if you target the mitochondria of neurons, surprisingly, ATP levels do not fall, but NADPH levels fall dramatically, right? So the phenotype that we are observing, this neuronal cell death, cannot be attributed to changes in ATP, in the bioenergetics, but rather in the redox homeostasis, mostly because NADPH levels are being depleted. And of course, we're validating this. We want to test causality. What we did is that we knock out either PC of malic enzyme 1 in astrocytes. And remember, astrocytes, normal astrocytes, are protected against mitochondrial poisons. But when we knock out PC or malic enzyme, you clearly see that cells start crashing, and again correlates with decreased levels of NADPH. We are now validating this in neurons overexpressing malic enzyme 1, not just in vitro, but also in vivo. I'm showing you a model that we're using that recapitulates one of the most severe diseases in humans, which is called Late syndrome, right? Those mice are knockout or a specific complex 1 protein. They have a shortened lifespan, as you clearly see, and a progressive and fatal neurodegenerative disorder. We perform metabolomics on the brain of those mice to confirm that redox homeostasis, mostly levels of NADPH and glutathione, are reduced in the brain of these mice, but surprisingly, metabolites linked to bioenergetics, such as ATP, do not change, right? The same thing that we saw in cells. So the idea is that can we overexpress malic enzyme, and the rationale for that is this, the overexpression of malic enzyme should restore NADPH levels and glutathione levels, and maybe that would be like a good therapeutic strategy. So we're working on that. We have preliminary data, not very successful yet, but we have a really reduced number of mice where we are injecting directly into the brain an AV associated virus containing the malic enzyme one, right? We start seeing some like phenotypes here. These are like really preliminary data. There is like a small trend towards like the malic enzyme 1 overexpression, reducing a little bit some pro-inflammatory markers in the brain, but we're still like working on that. So I want to end up giving you what is like the model, which basically is like under normal conditions, you inhibit PPP, the cells do not care because they still have function in mitochondria and they can produce a lot of malate, and then through malic enzyme 1 they produce NADPH, right? So cells are okay. But what happens when you target mitochondria? So if you inhibit mitochondria, the lethotransport chain, then what happens is that your survival might depend on how much malic enzyme 1 you have, how much NADPH you can extract from it. So you have like a lot of malic enzyme 1, you have these adaptive mechanisms in the form of PC and ACLY that is going to give rise to malate, and then the malate can be transformed into NADPH. But if the malic enzyme 1 levels are very low or these adaptive mechanisms do not work, then the cells crash because they've been depleted of NADPH, ROS ratio oxygen species skyrocket, and then eventually this is going to kill the cell. So finally, I want to acknowledge my group. This has been mainly the work of Mauro and Lucia. They've done a terrific job, and also like I want to thank like all the collaborators, both nationally and internationally, the funding agency. Also thank you, the audience, and I will be really happy to take any questions you might have.
So thank you very much. Remember that you can make your questions in the chat if you want, both in the chat of Zoom and also in the chat of YouTube. Thank you very much, Eduardo. Susana, you are muted. If you want to make any question, we cannot hear you.
Oh, sorry. Thank you very much, Eduardo. I was saying things, but I had silenced my microphone. Thank you very much for the interesting talk. I don't see any questions in the chat, but I will start with a question. These alternative pathways to generate malate, taking into account the effects on ROS and on survival, would you consider that these are adaptive mechanisms, or could also have any detrimental consequences for cells?
Well, definitely they are beneficial in the context of mitochondrial dysfunction. For instance, like there are like cells that, because they naturally have abundant expression of these proteins, such as astrocytes, they are more naturally protected towards like these mitochondrial insults. So maybe like what you want to say is that, okay, if we consider this as a strategy, we want to manipulate the levels of malic enzyme 1, for instance, overexpressing malic enzyme 1 in neurons that they naturally do not have like this abundance in this protein. Would this be deleterious? So the answer is it could be. So, but this is always like this therapeutic window. Is there like, what is worse for a neuron? To have a lot of malic enzyme 1, or just die because they don't have it, and then when you try mitochondrial dysfunction, they are quite vulnerable, right? So there's always like this trade-off. And at this point, we would favor that, okay, we can manipulate, or if we are able to manipulate the activity of malic enzyme 1 in certain contexts, that will be beneficial, or at least the benefits outweigh the detrimental effects.
Yes, understand. Yes. I found interesting that the effects in neurons, that they do not affect, they have low levels of these enzymes, right? But this does not affect the bioenergetics of the cells, but rather the redox homeostasis. Have you, apart from ATP synthesis, checked the activity of complexes, the different complexes in neurons?
Yeah, like for us, this was really shocking. Like not seeing any decrease in ATP when we inhibit mitochondria was really surprising. We repeated these experiments over and over to confirm that was the case. Also, what we did is that we collaborated with another group in France, and they have like these ATP biosensors, so they could measure ATP real life. They transfect neurons, and then they put these drugs that target the mitochondria, and they follow the ATP levels. They've done it not just in neurons, but also measuring ATP in the sum of the neurons, also at the synapses of the neurons, and as a whole. And again, the experiments are crystal clear. They don't see any decrease in ATP levels in these neurons.
There is a question in the chat that is also related to this discussion. It's from Antonio Martinez Ruiz. He says, fantastic work. We have learned from Juan Pedro Bolaños and Ángeles Almeida that neurons rely very much on PPP for NADPH production. That would explain why they don't have so much ME1, but why are so sensitive?
Yeah, this could be. Yeah, like Bolaños have done like this fantastic work, as they said, in the metabolism of neurons, mostly this intermediary metabolism. They've shown that the fate of glucose in neurons is mostly to fit into PPP, right? When we claim neurons are oxidative cells, well, they oxidize pyruvate, not the glucose, right? Actually, if you force them to perform glycolysis, and this is at the expense of PPP, neurons also start dying. So yeah, as Antonio is mentioning, probably is true, is they rely so much on PPP, the oxidative drug of the PPP, as a mechanism to produce NADPH, that they have neglected these other like alternative enzymes, malic enzyme 1, but also IDH1. So that could be, yeah.
We have also a couple of questions from the YouTube chat. Bella Mora Romero asks you, why do you use PRC to inhibit the complex 1 instead of other drugs such as rottenham?
Well, we mainly use pyridicidin because it's more selective towards complex 1. For instance, rottenham it's been shown that affects also the acetylation in microtubules, and this is killing, for instance, neurons. And actually, the experience I think was done in neurons. Rottenham is killing neurons not because it targets complex 1, because it targets the microtubule formation, so it has like more side effects. Now the scientific community have shifted towards the use of pyridicin instead of rottenham. You see that rottenham is mainly used now in sequence experiments where you have like, intermitochondria and you want to run like really short experiments. But if you want to work with culture cells, I would suggest that you start using pyridicin A.
Okay, and Silvia Mora tells very interesting talk. Thank you. Is there any evidence of that this pathway is involved in neurodegenerative diseases such as Alzheimer?
Well, mitochondrial dysfunction, yes, it correlates really well with not just Alzheimer, but mostly Parkinson, I would say. Like it's been like this really a fantastic job by Patricia Gonzalez Seville. They have like this complex 1 specific knockout in dopaminergic neurons, and they can recapitulate most of the clinical features of Parkinson. So there's a strong link there. And it's always been like rottenham is a pesticide and correlates really well the areas where rottenham is used, like the prevalence of these neurodegenerative diseases such as Parkinson also are really high. So that's it. Like in terms of like the pentose phosphate pathway or the malic enzyme, this is not well known. Actually, I couldn't find like really good papers linking like this enzymes to these diseases. So this I completely don't know.
Okay.
Sorry. Yeah.
No, Susana.
There is another question in the chat from Alberto Vázquez. He says, "Thank you, Eduardo, for this very interesting talk. Have you studied what changes occurs in the morphology of mitochondria or mitochondria crista when cells use these optional pathways?"
So this is very, very interesting question. Well, the short answer is no. We haven't tested that. We haven't analyzed either the ultrastructure or the dynamics of the mitochondria in terms of fusion and fission. But I think you're right. We should test it, and maybe because we still don't know how these pathways, these alternative pathways, like kicking, like why they. We think we haven't seen increase at the level of the proteins. So we think it was more like the fluxuses, like the metabolic fluxuses. But it could be that this relates to different conformation of the mitochondria. So it's something that definitely, yeah, we should check.
Related with these questions, Gonzalo Ranz asked first, he says, great talk. Do you observe mitochondrial damage and increased number of mitochondria with PRDC treatment?
Repeat again, sorry?
Do you observe mitochondrial damage or increased number of mitochondria with a PRDC treatment?
We haven't like observed any increase in mitochondrial mass upon PRDC treatment, and keep in mind that those are like short-term treatments, like 48, maybe like 20, like 3 days maximum. And in terms of damage, we first should like define what damage is, and by damage maybe it's looking at electron microscopy, and if they don't look okay, if they are swollen, and all these kind of features. We haven't tested that. Maybe like that could be interesting.
And Erika Fernandez Bizarras says, nice work. Have you tested if ROS scavengers can also recover cell viability?
Yeah, we did, like both NAC, but also to greater extent glutathione. You can supplement cells with glutathione molecules, which is reduced. And this is a permeable version, a cell-permeable version with glutathione. It's terrified. So we can process the cell membrane and goes directly into the cytosol. We can nicely rescue cell survival. We are trying this on neurons, but apparently like the levels of glutathione that we can put in normal cells, like we use like 2 millimolar glutathione, cells tolerate it really well. It looks like neurons, they don't like it that much, so we are lowering the doses and repeating those experiments in neurons.
And I have a curiosity. You have said that you're not interested in cancer, but I am interested in. and of course, these pathways could be part of the adapting mechanisms of cancer cells to cell death. So how do you envision the applicability of your research to cancer treatment?
Maybe I should rephrase. I'm highly interested in cancer. Actually, I have like a really big project like related to cancer and metastasis. But it was more like narrow, like in the context of this project. Use cancer cells just to prove like this was a general phenomenon. But yeah, as you mentioned, like there's been like nice papers establishing that enzymes that produces NADPH are beneficial for melanoma cells to metastasize. It looks like when cells are trying to metastasize and enter the bloodstream, they start facing a lot of oxidative stress. So cells that do have more of these pathways, and it's been mainly demonstrated by increasing the pentose phosphate pathway and also the 1-carbon metabolism pathway through LDH1 L2. So those cells, and in the context of melanoma, those cells are really protected from oxidative stress, and they can metastasize more. So it's like a huge link between these NADPH generating enzymes and tumor progression and metastasis. So it's another angle of investigation. Yeah.
This is a good target, not to avoid metastasis. Yeah. Okay. Thank you very much. Do you have any more questions, Susana? We don't have more questions in the YouTube.
No, in the chat either.
Okay,
no.
So thank you very much, Eduardo, for this fantastic talk. Thank you, Susana, for organizing this meeting. And
thank you.
Thank you also for all the people that is all the Mondays that we have these webinars is attending and making questions and making these webinars more active, and this in particular has been really active in questions, and we will see you in the next webinar, and thank you for attending this. Thank you.
Thank you.
Bye. Perfecto, ¿no?
Muy bien.